Mitosis is the stage in which the cell grows and DNA is replicated in preparation for cell division. It's comprised of 3 substages: G1, a period of normal cell growth and function, S, where chromosomes are duplicated, and G2, a final growth period before division. During mitosis, the cell divides. There are 3 checkpoints, most importantly, G1, which cells must pass through to go on to M phase. Cells that do not pass through this checkpoint will not reproduce, or they must do other things before dividing, such as go to the G0 phase to be repaired before moving on. G2 checkpoint makes sure that DNA is completely synthesized and the cell is ready to divide. During the M checkpoint, if spindles are not properly attached to chromosomes, cell division is halted until they are fully formed and functional. Mitosis allows cells to reproduce, to repair and replace damaged or worn-out cells. Prophase. Chromosomes condense, nucleoli and ribosomes disappear, and the mitotic spindle begins to form. During prometaphase, chromosomes condense even more. Kinetochores are developed by each chromatid and attached to spindle fibers. During metaphase, chromosomes line up in the middle of the cell or at the metaphase plate. They are moved by motor proteins. During anaphase, the shortest stage, proteins are attached to chromatids, are cut apart. Sister chromatids are pulled to opposite poles of the cell. Newly formed daughter chromosomes are pushed and pulled by motor proteins along kinetochore microtubules. The cell also expands. During telophase, 2 nuclei form, and the nucleoli are recreated. A new nuclear envelope forms around each set of chromosomes. Microtubules break apart and mitosis is complete. Mitosis results in 2 genetically identical daughter cells that have the same number of chromosomes as the parent cell. Meiosis is the phase of cell division in sexually reproductive organisms in which half the DNA is provided from one parent and half from the other. Meiosis allows genetic diversity in populations. During meiosis 1 is the reductive division. Prophase 1, DNA synthesis occurs. Nuclear envelope begins to break down. Nuclear chromatins start to condense into individual chromosomes. Each chromosome is copied and sister chromatids are connected via the centromere, via protein complexes called cohesins. Homologous pairs of chromosomes bind in synapses and are called tetrads. Crossing over may take place, where large sections of chromosomes are transferred between non-sister chromatids. Points of contact are called chiasmata. Centrosomes are also duplicated. During metaphase 1, tetrads move along microtubules and line up along the metaphase plate. Alignment of tetrads are completely random. During anaphase 1, microtubules attach and kinetochores pull chromosomes toward opposite poles of the shell. Sister chromatids remain bound. During telophase 1, complete haploid sets of chromosomes are at each pole. Nuclear envelopes form around each set as they decondense, and cytokinesis begins. Meiosis 2 is the separation of sister chromatids. During prophase 2, spindles form, the nucleolus and nuclear envelope break down, and there is no exchange of DNA between sister chromatids. During metaphase 2, chromosomes align in the middle along the metaphase plate. Anaphase 2, centromeres of sister chromatids separate, sister chromatids are pulled to opposite poles by microtubules, and each sister chromatid is now considered a chromosome after separation. During telophase 2, chromosomes uncoil, the nuclear envelope begins to reappear, and spindles disappear, and cytokinesis then splits the 2 cells into 4 daughter cells, containing a haploid set of chromosomes. Meiosis results in 4 non-identical daughter cells, with half the number of chromosomes as the original parent cell. While mitosis and meiosis are both processes of cell division where genetic material is shared between parent and daughter cells, there are some major differences. During mitosis, 2 genetically identical daughter cells are formed, while during meiosis, 4 non-identical daughter cells are formed. Mitosis has a function in cellular reproduction, while meiosis' function is in sexual reproduction. Mitosis is a type of asexual reproduction, and meiosis is a type of sexual reproduction. Crossing over does not occur in mitosis, while it does occur in meiosis. There is 1 division during mitosis, and 2 during meiosis, and the same number of chromosomes are in the cells after mitosis, while half are after meiosis.